out here, lateral on the brain. So now I'm going to go to my neck muscle. I come out from lateral. Now that big giant pyramidal neuron sends an axon, passes down through midbrain, pons, medulla, and what happens when it gets to the medulla? Crosses over to the other side. Now where is it? It's in the lateral funiculus now, right? Okay, but can you be more specific? Yes, you can. Notice that in the lateral funiculus here, the body is actually separated out into sections. So all the axons that are headed down to the lumbosacral plexus are out more lateral. And all the axons that are going to be in the cervical lumbar plexus are more medial. And so you would know when you look at a spinal section some more information if you had this image in your head. What would happen if the regions more medial in the lateral funiculus were damaged? You're more likely to damage axon tracts that are controlling cervical region muscles, things in the neck and the upper torso. As you move more lateral, those axons in the lateral funiculus now are right. They're lower muscle groups, right? So think about it from the neck to the tail as medial to lateral within the dorsal funiculus. Okay, so a little bit more detail. Now we're going to do this with the sensory also. Can you see it? We're going to do it with the sensory also. So we're going to move. We're going to move. The same phenomenon works here in the spinal thalamic pathway from cervical to sacral. So you move out here. This one is the spinal thalamic. We'll do that one in a minute. But you can see here in the dorsal funiculus, this is where all the really detailed sensory information is. It's also cervical to sacral, but the other way. And I'm going to give you some numbers on that one. Okay, take it down to T6 and then take it below T6 with the funiculus and the cuneatus. All right, just things to look forward to.
Why do you think it's oriented like that? Like, sir, why do you think it's oriented like that? Going in closer, you think it's importance of like survival? I mean, if your neck or over your legs, I'm talking about like lumbar sacral, going in like that.
Why?
It just is like that.
It's organized. It's definitely organized. Okay, so premotor cortex. So out of the primary motor cortex now, let's move into the premotor cortex. So I'm just moving forward now on my section, on my slide section. This premotor cortex has all kinds of connections. Y'all, this is not for you to memorize. You're going to take a neurobiology class one day in your life. And I like to throw things in here because this is just all outline. And one day when you learn the details, this is the kind of thing you can look back at and say, uh-oh, here's the pieces. Okay, and then you can, whatever the horror of the detail is you're being given, you can use this as kind of outline. How does the primary cortex, how does the premotor cortex hook up, and what does it hook up with? So this is why I listed these for you. Let me tell you what track tracing does, because I've done quite a bit of this in my lifetime. There are all kinds of ways to track neural pathways. Most of them are one synapse track tracers. You inject the stuff in there, then you kill the animal, you section the brain, and you can tell it fills up one pathway. It doesn't cross the synapse. It goes from here to there. But there are other things that will actually cross synapses, and those are fantastically expensive. But this is how you get information like this. Neuroanatomists have, for years, injected track tracing molecules into brain areas of animals, and then killed the animals, and then looked at the brain to see where those pathways go. And there's good news and bad news in the nervous system. What I'm teaching you over the next 2 or 3 class periods is the absolute bare bones skeleton. This area of the brain controls this part of the body by a pathway that looks like this. But if you put a track tracer in somewhere, in the eyes and track that stuff, or in the brain and track that stuff, the thing that overwhelms you, I'm speaking from personal experience, is it appears that from wherever you are, you can get to wherever you want to go. Certainly, there are dominating pathways. But all this stuff in your textbook about regions that light up, in functional MRIs, and so we know this is what's happening in this area. What they don't tell you is that there are always outliers in these pathways. They don't even publish, but if you look at the data, you can see them. The connections in the human brain are absolutely mind-blowing, staggering. I always say that on this picture. Because the students see this and they go, you've got to be kidding me. So you guys don't know what the subliminal area is. You have some idea what's in the parietal cortex. All kinds of sensory stuff there. We looked at it on the first picture. Language stuff is there, etc. There are direct pathways from the premotor to the spinal cord. I thought you said that the primary pathways for motor control come from the precentral giants. Y'all see what I mean? So when somebody gets real dogmatic with me about a pathway, I say, okay, what you need to say is that's most of them. Because you can get to all kinds of places in the body. So that's why I just wanted to point that out. I like to say that with the track tracing stuff. That's where all this information came from. Okay, so primary and premotor cortex, and you guys know that this PMA, this premotor cortex area, is the area that is the association area for muscle contraction. That is a fair way to think about it. The premotor cortex. All right. Now, the next one is the frontal eye field, and I like to use this one to introduce just a little bit more. Just pull them along a little bit more. Again, your textbook is not going to take you here, but stuff like the pontine nuclei, the mesencephalon, the superior colliculus, the substantia nigra, those things are all coming in your life in the next 2 class periods. So if I bring them up here, they don't seem so foreign to you. All right, reminder first. If it's a cluster of neuronal cell bodies, does it look white or gray? White. It looks
Gray.
It looks gray. Why?
You can't be myelinated.
Okay. All right. So let's. Okay. So if you look at brain sections or spinal cord sections, this is obvious. The areas where the densities of the cell bodies are. All right. So what do I call those dense cell body areas where the clusters of neurons are outside the brain and spinal cord? Ganglia. And what do I call them inside?
Nucleus.
Yeah, I call them nuclei, which is a bit confusing because a nucleus we think of as, yeah, where the DNA is. That's not what a neurobiology person means. When they say nucleus, they normally mean a cluster of thousands of neurons that do something. So when you see caudate nucleus here, the nucleus of the substantia nigra, the nuclei of the pontine reticular formation, the nuclei of the superior colliculus, you're talking about major groupings of neurons that have connections that we now know are really, really important. For example, you're going to learn 2 pathways that come out of the midbrain that project into the forebrain. One of them will come out of substantia nigra neurons. And it is a pathway now that we know a lot of details about in the way that it controls Parkinson's disease. And so some of you may have seen some of this, pay attention a little bit in your life. Michael J. Fox has actually had some stereotaxic surgery done to his brain to try to alleviate some of the problems with Parkinson's. And many other procedures have been done in order to try to help with the neurotransmitter levels and stuff like that. Well, that information, the reason they know where to go is because of track tracing studies connecting these nuclei. And so I'd like for you to be able to connect some of them. Even though your book doesn't do it, I would like, as anatomy students, your teacher has some neuro in his background. I'd like for you to have a little bit of this in your background before you leave me. So this picture helps me bring up some of these words. You can see here the frontal eye field. And remember what I said by that was, you're going to get the impression that humans are visual creatures. If we were to do track tracing things like this in echolocating bats, we would get some different pathways because they're auditory creatures. Their inferior colliculi look different than their superior colliculi compared with humans or rats or sheep. or deer or cows, whatever brains we're going to use in the lab. So we're visual creatures. The frontal eye fields connect all of these different areas. So you can see that the frontal eye field here, just keep a list, and I'll say just a brief word about each. We'll pick them up as we get to them. Notice that the frontal eye field here has a connection to the caudate nucleus. Now the caudate nucleus is part of the basal nuclei. This is the third piece of telencephalon. On your outline it says cerebrum, white matter, basal nuclei. The caudate nucleus is one of those basal nuclei. Notice that the next connect, I don't expect you to be able to connect these yet. I just want to bring the words up so that when we get to them I can say, does that have anything to do with vision? And you're going to go, yeah. I'm going to say, how do you know? Because the frontal eye field is connected to it. Okay, so just a list. So here's the second one, the substantia nigra. The substantia nigra is a midbrain nucleus. And it is important, the substantia nigra is critically important in regulating skeletal muscle contraction. Now wait a minute. Skeletal muscle contraction is controlled from the precentral gyrus. And now you're telling me the midbrain is involved? The substantia nigra of the midbrain? Yes, that's what I'm saying. Why is the Parkinson person shaking? The answer is because these neurons of the midbrain die for reasons that we don't understand. And if you lose them, you lose skeletal muscle control. Let me show you this first outline picture here that we have. You see the basal nuclei are connected here? So let me show you some things you do know. Let me show you some things you do know, and then we'll go back to the picture and keep talking about the things you don't know. You already know that the cerebral cortex connects by way of pyramidal corticospinal tracts to lower motor neurons, right? And you also know that of the other motor systems, the tecto, reticulo, rubro, vestibulospinal, these guys, for general human biology, are relatively insignificant. They do not compare in importance to the pyramidal tracts. So, I'll say it again. You think move, you think pyramidal tract. You know they cross, and you know something about their pathways now. Okay, so some of these others are going to come up along the way. We haven't talked about the brainstem reticular formation yet. We haven't talked about the superior colliculus yet. That should ring a bell. We haven't talked about the basal nuclei yet, but we are going to talk about them. They're coming up. Okay, now, the reason I went back to this was to talk about, where was I? Go back to it again, remind yourself. Yes, the substantia nigra and the caudate nucleus. Okay, so the substantia nigra is part of the pathway that I'm going to teach you in just a second, so show it to them on this one first, these overall slides. Okay, so notice that the basal nuclei are getting connections from the cortex and sending informations to it. So I'm going to emphasize the second one, the basal nuclei and the projections up this way through the thalamus. Y'all are going to fill in blanks here. You're going to get a globus pallidus connection here, putamen and globus pallidus connections here, and you're going to put those into the ventral anterior and ventral lateral thalamus, and you're going to take those into the cortex because this is the pathway that they lesion on Michael J. Fox. And I'm going to show you why, where these neurons are and why they lesion them. Okay, now the reason I went back to the basal nuclei here is because these guys are the ones that get information. It's not on this picture, but they get information from the midbrain. So you have to learn the pathway from the substantia nigra to the basal nuclei in order to understand why a Parkinson's person is tremoring whenever they have damage to the midbrain. Okay, so come back to that, just to show you that picture. All right, so just a list: caudate, substantia nigra, pontine, I'll hold on that one, and this one, the superior colliculus. The superior colliculi are the visual reflex centers of the midbrain. So you can see there's a direct projection here from the frontal eye field into the superior colliculus. So in physiology, we're going to talk about the details of the saccadic eye movements. The It's one of the most fascinating things about visual physiology to me is I know when light hits the cell that it bleaches rhodopsin. So that is a chemical reaction. In order for that neuron to send a signal, it has to reconstitute the rhodopsin. You bleach it, and you go down a long pathway, 18 different molecular steps, and you send a signal to the brain. But then you've got to rebuild this stuff, or you can't use that cell again. So if you just stare at something, and you just look at it, you bleach the rhodopsin out of that thing, and you would be blind. But none of you do this, right? When you look at things, when you stare at things, they don't disappear. Why not? Because your eyes undergo these little tiny fractional twitching motions, imperceptible, where they move the light photons around on neurons in the vicinity, allowing the neighbors to reconstitute the rhodopsin while you're looking at things. The best way to illustrate it is to stare at a star at night, because it's a little more difficult for your eyes to move because the object's so far away to move enough for you to do your saccadic motions. You look up at the star and it disappears. How do you get it to reappear? Have y'all done this before?
You look away.
You look away, look back, and there it is again. Yeah? So that's that phenomenon. And the superior colliculus is involved with this. It's tracking movements as you're following something. This is the area of the brain that does that. Okay, so just words here. I don't expect you to know the connections of the frontal eye field, but I wanted you to hear caudate. I wanted you to hear substantia nigra. I wanted you to hear pontine and reticular and superior colliculus, because I'm going to talk about all of them. All right, so frontal eye field says we're visual creatures, and look, there's a lot of connections for the visual pathways. All right, now, the last one for the day is the speech area. You guys learned from me that, so we're doing the order. You got precentral gyrus, you got frontal eye field, and now you get Broca's area. So if you look at the first slide we looked at, this is the order that we're going in, same order as your book. And so here's your precentral gyrus right here for controlling muscle contraction, and here's Broca's area. Now, Broca's area is said to be a motor speech area, and there's all kinds of really interesting observations to be made about it. The first one is, it's only on the left side of the brain. So human brains are fascinating in part because of the unequal distribution of functions. Everything I have said so far today has been, this is what the brain does. The left side goes to the right side goes to the left, but very famous Nobel Prize winning series of experiments done by a neuropsychologist named Roger Sperry many years ago tested the functions of left and right brain. And the foundation for what he did, you're going to learn from me. Because Roger Sperry knew, and the neurosurgeon that worked with him knew about visual pathways. They knew that the medial side of the retinas cross information at the optic chiasm over to the other side, and the lateral sides of the retina stay home. They knew this because they knew their visual pathways. And they had a group of patients who had reached a point in their lives where they would do anything to survive. Patients with grand mal epileptiform seizures that were occurring at regular rates were willing to have anything done to try to prevent this. And I use this as an illustration with you guys. You want to see what a brain looks like when you're using all of it? That's what it looks like. It's not pretty. And so the neurosurgeon went in on these patients and cut the connection between the left and the right brain. A structure you'll see in the lab next week in the brains that you're going to look at, called the corpus callosum, the connecting axon pathway between the left and the right brain. They cut it, physically separating the left from the right brain. So as the seizures would begin, almost always on the right side of the temporal lobe, they start, and then they spread from that right temporal lobe across the brain, completely making the person completely dysfunctional and deadly. And so after they cut it, the intensity of the seizures went away of all the patients. They were all helped. And if you talked to them, you couldn't tell anything was different. It was a miraculous set of human experimentation. Cutting the corpus callosum, separating the brains, saving their lives. Now Sperry said, there's got to be something more here because the visual pathways cross from the middle, stay on the same sides. So they ran a series of experiments on them, and the result of that is in your textbook. A lot of stuff is on the left side only, where a lot of other stuff is on the right side only. And you know this. If you call somebody a left brain person, what did you just call them? You say, well, you're left brain. Okay, let's start with the other one. You said, well, you're a right brain person. What did you mean by that?
They're very artistic-minded.
Yeah, everybody knows this. So the aesthetics and beauty and talent and creativity, it's on the right side. And all the math and logic is all on the left side, right? Where did that come from? It came from track tracing. It came from Roger Sperry's stuff. And one of the things that they know, that they discovered in this, was the speech stuff. Now, we didn't really need that today, because today we have functional MRIs. And you can ask people to do things, and you can watch their brain to see what areas are lighting up. And so, one of the things that came out of all that, left and right brain stuff, was this. Broca's and the speech areas are primarily on the left side. But it's way cooler than that, because in humans, handedness is related to this. So, most humans, 85% of the human population, is right-handed. 15% of the human population is left-handed. Of the right-handed people, the vast majority of them, different estimates on this, but it's a big number, are what you would consider to be left-brain individuals. They're left-brain dominant. In fact, when I learned neurobiology, that's what it was called, cerebral dominance. So it's not called that anymore. It's not politically correct to say neuroscience anymore that way, because the right-brain people were offended by that. So if you're left-handed, then you'd be right-brain dominant. So where would the speech areas be for a right-handed person? On the left side, okay. Where would the speech areas be for a left-handed person? On the left side too, yeah. Although not all of them. So there are left-handed, about half of the human population with left-handedness have speech areas on the right side as well. So when we say it's left side, you just mean most of us. That's what you mean. And there are two big ones, Broca's and Wernicke's, and they're connected by a tract of axons called the all-famous, here it is, the arcuate fasciculus, connecting Broca's and Wernicke's area. Now Wernicke's area, as I said here, is involved in sounding out unfamiliar words. Another way of saying that is Wernicke's area here in the superior regions of the temporal lobe is primarily responsible for understanding speech, as are some of these other areas very nearby. Understanding language and coordinating aspects of language. They're all right along that posterior rim of the temporal lobe. That's where all of the I understand you stuff is. And that information is connected to this motor speech area. That is, I understand you, now I will respond to you. Language, written, spoken, whatever, up here in Broca's area. So the Broca's area is this motor speech. I'm going to talk now. And if you interrupt this arcuate fasciculus, a stroke or something else damages, these are fascinating patients. And I've seen some videos. They understand and they can speak, but putting the two together is problematic. So oftentimes they will create in their speech what's known as a word salad. It's very frustrating for them. They talk, and they'll start to say words, and they know what they want to say, but it comes out. The word order is not, they call it a word salad. It's not, it's like all the words are just tossed up. And when they hear it, they know it's not right. And so they stop. And they'll say it again. And it takes them several efforts without an arcuate fasciculus intact to actually put the words in the right order. So language is fascinating. It's on the left side primarily in Broca's and Wernicke's, but there are also other areas very close by that are involved in language skills. Okay, we did precentral gyrus, premotor area, frontal eye field, and Broca's. Okay, so when we pick this up next time, we'll continue to move down through the brain. Next week's lab will be brain anatomy.